Well, welcome back. This time it's about energy transformations. I probably should have warned you at some point that biology is really about understanding how chemistry and physics influences the natural world. So a lot of students think they're going to get an easy run with biology, but not always the case. So in this one, we're looking particularly at energy and energy sources and how energy is moved through living systems, living things. So you can be an autotroph or a heterotroph, and therefore you need to be able to do photosynthesis or chemosynthesis. Then you need to use that energy. So cellular respiration, either anaerobic or aerobic, releases energy. And of course, what's that energy doing? So, excuse me, I'll blow my nose. Autotrophs are organisms that are able to make their own organic materials. So we know of plants. Plants are a clear example. They take light energy and use it to combine carbon dioxide and water to create glucose. And of course there are also things that live in deep sea vents and around volcanic outlets like Rotorua, where they use the chemicals that come off those sort of places to chemosynthesize. They use the chemicals to create a form of glucose. We're heterotrophs. We've got to eat stuff. So we've got to eat stuff to get the energy from it. So it's inefficient for us in that sense. More inefficient for you carnivores than it is for me, the vegetarian, but we won't push that barrow just at the moment. Well, I'd like to. Cheers. Excuse me. So, clearly most autotrophs are plants, as I've already said, and the sense here is to trap light energy, or in the case of chemosynthesis, chemical energy, and convert it into a stored form of energy. So glucose is stored chemical energy. So the outcome isn't directly useful, but it's a source of energy that will be useful. So we're looking at lots of transformations and transfers of energy here. So photosynthesis, critical for life on the planet. Clearly it allows us to have glucose, so I can eat a plant and get energy. It allows plants to eat the glucose they make for themselves and get energy they need. But you'll see at the end here, there's a really bad waste called oxygen. So photosynthetic ability really did change the planet from one with low oxygen levels to one with much higher oxygen levels, and allowed for more and more complex life to evolve and develop, and so it allowed us to exist. So 6 carbon dioxide atoms react with 12 water atoms in the presence of light and a catalyst called chlorophyll, which is the pigment which gives plants their green colour. There are a whole lot of other pigments as well, but chlorophyll is a major one. The outcome is glucose, C6H12O6. There's a bit of water left over, and there's oxygen left over. So these are wastes. So really useful wastes, obviously. And this all happens in the chloroplast. So the chloroplast, again, I put myself in the wrong place, haven't I? Chloroplast is an ancient prokaryote, we believe, that is actually living in symbiosis with the eukaryote. So it's what's called endosymbiotic, so inside the cell. It gets protection from the cell, obviously, and lots of food the cell creates, but it also creates lots of food for the cell. So there's a two-way thing going on here, symbiosis. Why do we think this? Well, it's got a biphospholipid layer around the outside. It has DNA occurring in plasmids, which means it's capable of self-replication. So a plant cell, or plant cells and plant tissues like leaves exposed to light end up having lots of chloroplasts in them. Plant cells like roots don't have any. They're no use. So that replication is very important. It occurs in 2 stages. I'm going to go into a little bit of detail of chemistry here that you don't really need, so I'll go through it quickly, just so you're aware of what goes on. So there's 2 stages. The first is what we call light dependent. So that light energy is required to actually split the water atoms. So those 12 water molecules become 24 hydrogen ions and 6 oxygen gas molecules, 6O2, that waste we see at the end. And the 24 hydrogen ions are then used to carry the energy. So they're donating protons. Won't get too much of the chemistry here, but there's a whole lot of biochemistry happening here. Oops, I went the wrong way. In the second stage, so it's really quite complex, doesn't need light, so it can actually occur at night, and some plants actually deliberately do that at night to save water. In this case, all that hydrogen is used with the carbon dioxide to create, well, you can see a series of different glucose or carbon-based things. We've got a 5 carbon chain here. There's some CO2 coming in. Got 3 carbon bases with a phosphorus that exchanges that for energy. It gets quite complex. But in a sense, what we end up with is this thing called a G3P. So it's got 3 carbons, and 2 G3Ps create a glucose. And so the outcome in time will be glucose. G3Ps are also used to make sucrose and starch. So, what's going on? Light dependent occurs in what's called the stroma of the cell. The inputs are water and light energy. The outputs are hydrogen ions, oxygen as waste, and ATP for use later. There's no energy created yet. In the grana within the chloroplast, those hydrogen ions and their carbon dioxide are used to create that glucose, well, 2 G3Ps, and we have some wastewater left over. But more importantly, we now have 38 units of what's known as ATP, adenosine triphosphate, bound up inside this glucose molecule. That's the stored chemical energy. ATP is sort of the energy currency of a cell. Now, the opposite to that is a thing called cell respiration. This occurs in mitochondria. So in cell respiration, we take the glucose molecule, we burn it, essentially, with oxygen, hence the heat is created. This releases 6 carbon dioxide and 6 water as waste, but importantly, it releases the ATP stored in the glucose. So now the cell has up to 38 units of ATP. Well, probably it's not that efficient. You never get 36 to 38 ATP, but potentially there are between 36 and 38 ATP units available to the cell. And that energy is what the cell then uses to build all the other stuff it does and to run itself. And all organisms use ATP and the phosphate bonds that are formed. So ATP can be a MP, a DP, and an ATP, where the M means mono, the D means di, 2, and tri, 3. So adenosine triphosphate, 3-phosphate. But that's a bit too much chemistry already. This occurs essentially in 3 stages. So the first can be, well, we'll talk about anaerobic respiration first, but it's the same stage. So if there's not much oxygen around, the organism is actually able to carry out fermentation or anaerobic respiration. So it does this without oxygen, so anaerobic, no oxygen. But it doesn't go past glycolysis. So in plants, so we get a little bit of ATP, because we get a couple of units of ATP, but you get a lot of alcohol or CO2 built up. Now, we use the alcohol or CO2 in our food industries. So obviously we get some plants, we cause them to respire anaerobically, and they produce a whole lot of alcohol as a byproduct or as a waste. Bacteria often produce lots of CO2. Now, it can be used also to produce alcohol. In animals, we get lactic acid and CO2. Now, all these wastes, of course, are damaging to the organism, and they want to break them down as soon as they can. And they'll try and break them down with oxygen, obviously, because that's what we use it for. So athletes often have a problem with lactic acid and the build-up of that during events. So 3-stage process, glycolysis, can stop here and become anaerobic, or it can move inside the mitochondria. So this happens in the cytosol. We now move into the mitochondria and take the next 2 stages. And it gets quite complex in a biochemical sense. So mitochondria, again, has this double membrane, lots of plasmid loops, able to self-replicate. So I believe it also gives a piece of endosymbiotic evidence. So glycolysis, we've already described as glycolysis, but in this case of aerobic respiration, we've split the glucose molecule. That's what glycolysis means. So that becomes 2 pyruvate molecules, and 2 NADH, and I won't even bother trying to explain what NADH is, nicotine, which are hydrogen acceptors, and 4 molecules of ATP. So if there's enough oxygen, so if it's going to be aerobic, it spends 2 of those ATPs moving everything else across into the mitochondria. Hence this C638 thing. Krebs cycle. Krebs cycle occurs within the cell in the matrix, inside the mitochondria in the matrix, and the two pyruvates and two NADHs move into the matrix, and then they are changed into acetylcholine coenzyme A molecules, more NADH, and now some FADs, some FADHs, and we get a whole lot of CO2 produced at this stage, and 2 more units of ATP. Then we finish that off. I'm going to have to pause for a minute. The work's redone in electron transport. This occurs in the cristae of the mitochondria, and all those NADHs and FADHs interact with these things called cytochromes, and so they form bring a whole lot of ADP in, and the ADP get these phosphorus attached to them, and they become ATPs, and you can get between 32 and 34 here. And again, some more waste. These 6 molecules of water are produced. Now, there's a lot more in that you need to know at this stage, but I thought it's worth having that background knowledge. So, glycolysis occurs outside the mitochondria in a cytosol. It creates one glucose molecule. Sorry, it uses one glucose molecule. From that, it produces 2 pyruvates and a couple of NADHs and some ATP. Then we move to the mitochondria and onto the matrix. At this point in time, the oxygen comes in, and oxygen with the NADH and pyruvates create these 2 acetyl coenzymes, 6 NADHs, 2 FADHs, and some CO2 as waste, and a little bit of ATP. We haven't really produced much yet. And then the heavy lifting here is at the cristae, where those NADHs and FADHs are used to transfer hydrogens and create all this ATP. Unfortunately, I'm in the way, so 30 to 34 ATPs down here. And of course, there's more waste in the form of water. Not bad waste. We'll talk about metabolic water at some stage. This is a metabolic water here. We'll come back to it though. So, that's energy transformations. One more video for this first area of study. I'll be back.